Now I want to talk in a little bit more detail about the whole idea of alpha, beta, and gamma diversity, which was introduced in the last lecture, mostly because it's just sort of experience from the past that students have some issues with one particular part of this, the whole notion of beta diversity. So I'll get there in a minute, and alpha diversity. But let's talk about the easiest one first. What is gamma diversity? Gamma diversity is the species richness, or S, of the whole area. If you want to know what is the whole area, that's up to the choice of the worker. It can be the count of taxa in any different ecosystem, geographic area, province, whatever that you are considering. So, how would that work for our particular example here? We have the hypothetical species A through N, and if you count that, there are 14 of them, and so in this whole world we're considering, this shoreline area at some ocean beach there, the gamma diversity is 14. So gamma diversity, diversity of the whole, for us 14. What about alpha diversity? Alpha diversity is simply species richness within a single ecosystem or community or sampling block or whatever you're looking at. So you just count the number of taxa in that place. For our examples here, I've circled the number of species in each of the columns there. So recall, this is marked as ones and zeros, 1 meaning present, a species is present, and 0 meaning it's absent or not there. And so for the supertidal region, you count 10 species, species A through J, so the alpha diversity is 10. For the intertidal area, the alpha diversity is 7, and for the subtidal area, we just have an alpha diversity of 3. But what about beta diversity? This is the one that's a little bit more confusing, because it's a measure of richness between two ecosystems or communities or sampling blocks. So you do sort of the same thing. You count the number of taxa in a place, but then you want to compare that number to the number in another place. And normally these are two adjacent or next to places, next to ecosystems. There's an additive nature to it, such that you count the number of taxa that occur only in one of those locations, and then you compare it to the number of taxa that you count in that other considered location. So let's look at a quick example. If I want to know the beta diversity, and it's always phrased this way, it's the beta diversity between something and something else. So here, the beta diversity between supertidal and intertidal. First, I look at the number of species or the species that are present in both. Notice there's, for species F through J, there's a 1 in both of these columns. I've drawn lines sort of Xing them out. We don't consider these in beta diversity. Instead, we consider the species that are unique. And so for the supertidal, species A through E, there are 5 of them. They're unique. And then for intertidal, species K and L, those are the 2 unique ones. Beta diversity takes those number of unique ones from one community, one ecosystem, and adds it to the number of unique ones to the next. And so here, the beta diversity is 7. To compare then the intertidal area to the subtidal area, the only species that is shared between the two is species L. Draw a line through it. And then the number of unique species is 6 plus 2 equals 8. Now you could do the beta diversity between the subtidal and the supertidal, but they're not next to each other. And so some workers do this, but most do not. So it's not normally considered. But if you were to do it, I'm not going to even put a picture up for you, but then you'd count the number of species, look at the number of unique ones, and then it would be 10 unique ones for the supertidal and 3 unique species for the subtidal, and the beta diversity there would be 13. but as I said before, this is not normally used.